Hi, so I'll start today's class. Okay, okay. So it's 6:15. I'll start today's session. Um, Anyone who's joining later I can join whenever they are available. I'll accept the request. So let's start with today's session. Today is the seventh live session of this set of live sessions, basically, where we discuss some previous year assignment questions based on the course Biochemistry taken by Professor K. Subramaniam. And in these live sessions, we basically discuss some basic conceptual questions. We discuss the patterns of those questions and the basic concepts behind those questions, so it's easier for us to solve similar type of other questions. Okay, so we'll start this session with the first question for today. So the first question is, what methods will be suitable for the separation of individual proteins from a mixture containing the following proteins? Protein A with molecular weight 13600 Dalton and isoelectric 0.6, or protein B with molecular weight 43600 Dalton and isoelectric 0.6.5. Protein C, molecular weight 25,600 Dalton, isoelectric 0.6, and protein D with molecular weight 25,650 Dalton with isoelectric 0.4. Which one do you think is the correct answer among these four options? The first option says A and C can be separated using isoelectric focusing. The second option says B and C can be separated using gel filtration chromatography. The third option says A and D can be separated using isoelectric focusing. And the fourth option says A and D can be separated using gel filtration. Which among these four options do you think is correct? To answer this question, basically you'll need to have a little basic about these different types of techniques. What does isoelectric focusing do? What is the criteria? What is the principle of isoelectric focusing? What is the principle of gel filtration chromatography? All of these. So, would you like to take a guess or answer? I'll wait for 2 minutes. Anybody? I'll wait for 2 minutes. Anybody? I'll wait for 2 minutes. Anybody? Okay, so we'll discuss a little about the principle of both these techniques, isoelectric focusing and gel filtration chromatography, and then we'll move ahead. Okay. So the answer for this will be B and C can be separated using gel filtration chromatography. That is, B this one and C can be separated by gel filtration, because gel filtration chromatography, the principle works on size, correct? So these two have pretty much a significant difference in their molecular weight. So gel filtration chromatography, the determining factor is the molecular weight. So these two have pretty big differences if we compare the molecular weights. So B and C can be separated on the basis of the molecular weight. So gel filtration chromatography. A and D can be separated using isoelectric focusing. So if you see A and D have significant differences in their isoelectric points, like if for here A it is 6, for D it is 4. So this can be separated on the basis of the difference of the isoelectric point by the means of isoelectric focusing. Whereas A and D can be separated using gel filtration. A and D also have a huge difference in their molecular weights, like it is 1300, 13,600 and it's 25,650. So even this level of difference can be separated using gel filtration chromatography. Okay, so these two statements, this statement is correct. Okay, A and so one incorrect option was A and C can be separated using isoelectric focusing. So A and C, this is not feasible, not possible, because both are the same isoelectric points. See, Pi6 and Pi6. So these two do not really have differences in the isoelectric point. So A and C cannot be separated using isoelectric focusing. Now we'll discuss in detail about the two techniques, isoelectric focusing and gel filtration chromatography. Okay, so we have already talked about amino acids, peptides and proteins, and we have already talked about the structure of proteins, the primary, secondary, tertiary, all these structures. So if we talk about the protein purification process, so in order to study the action of the activity of the protein, the structure of the protein in detail, a researcher will have to separate it from other proteins in pure form. So we need the pure form of the protein in question, and the researcher should be able to use the techniques to determine its properties. So to purify a protein, basically, one usually will start with a crude extract of a tissue or a cell sample, correct, and separate the proteins within it into fractions. Now, it's given that the initial volume of the crude extract is relatively large. A researcher will typically apply a technique like ammonium sulfate precipitation to reduce the size of the sample and the number of proteins within it. Now with ammonium sulfate precipitation, one will exploit the differences in the solubility of the proteins in salt solution to obtain a fraction that is enriched in the protein of interest. So we have to basically separate our protein of interest from any other component. Now throughout the multiple steps of protein purification, the researcher must have available some type of assay for monitoring the presence of the protein. Now we have, if we are separating the protein, we should have an assay that will be able to give us a visual readout so as to monitor the presence of the protein. Okay, so that is basically to understand if the protein is present or not. And subsequent to the ammonium sulfate precipitation, investigators will typically apply column chromatography procedures to further purify the protein. So once the ammonium sulfate precipitation is done, investigators will then apply column chromatography techniques to again purify the protein. Now what is column chromatography? It basically can separate proteins based on their net charge at any given pH. Okay, so at a given pH, ion exchange chromatography will be helpful, or if it's based on pH, ion exchange chromatography can be useful, or if the principle of separation is there at the relative sizes, then size exclusion chromatography or gel filtration chromatography can be useful. And if it's based on the ligand binding specificity, then affinity chromatography will be helpful. Now the general principle behind column chromatography is illustrated here. So as different proteins will percolate through the column, they will be separated based on their physical properties. Okay, so the effluent fraction will contain the protein of interest and can be identified when we have an enzymatic assay. That will give us a readout. So as we said, we can separate the proteins, but how are we sure that it's the protein we separated or it's something else? So that's when the enzymatic assay, enzyme based assay comes into play. Okay. So what happens in ion exchange chromatography? In ion exchange chromatography, proteins are separated basically based on differences in the sign and magnitude of their charges at a given pH. Okay, so so-called cation exchange resins are used to fractionate positively charged proteins in a mixture, and these resins contain bound negative functional groups, as you can see over here. Okay, so in case of cation exchange resins will be used to fractionate positively charged proteins in a mixture, correct? And these, if you can see, So this is the solution mobile phase that is being passed. The resins will have a negative charge. So large net positive charge will be attracted towards the resins. Small net positive charge will be a little less attracted towards the resins, whereas the negatively charged proteins, net charged proteins will be repelled and they will elute fast. Okay, so these resins will contain bound negative functional groups, as you can see in this diagram. Okay, so that is the diagram. Now, anion exchange resins will be used to fractionate negatively charged proteins in a mixture. That's the opposite. Okay, so in anion exchange resins will contain, so in anion exchange resins will have bound positive functional groups on the resins, correct? And so that then the negatively charged proteins will get attracted towards the positively charged functional group containing resins. And with both these techniques, proteins having the same net charge as the resin move through the column relatively quickly. So if we have a negatively charged resin and there are negatively charged proteins that will be repelled by the resins and they will not be attracted towards the resin, they will move through the resin faster, through the column faster. Okay. Now proteins with a net charge that is opposite to that of the resin are retained and ultimately are released by adjusting the pH or salt concentration of the elution buffer. So proteins that will have a net charge opposite to that of the resin, like if it's a negatively charged resin, the positively charged proteins will be retained, okay, and will be released and they'll be retained. Now once the everything else has passed out or has eluted, the positively charged proteins can be ultimately released by doing any kind of adjusting the pH or salt concentration in the elution buffer, and then those will get attracted towards the negatively charged resins, and everything all the positively charged proteins that were being attracted such heavily before will get released and be eluted out. Okay. Okay, so what happens in size extrusion chromatography? So in size extrusion chromatography or gel filtration chromatography or gel permeation chromatography, what happens? The proteins are separated based on their size or their molecular weight. So they need to have different molecular weights in order to be resolved properly and separated. So the resin used in size extrusion chromatography will be uncharged, okay, but it will contain pore into which small solutes proteins may be able to penetrate. So even though the resin will be uncharged, there will be small pores, uh, and inside the small pores, small proteins or solutes will be able to penetrate and go inside. So for this reason, the largest proteins move through the column the fastest, whereas small proteins are retained longer. Okay, so size extrusion chromatography in the presence of standards of known molecular weight can be used to determine the approximate molecular weight of an unknown protein. So the resins here in this case will be uncharged. However, the resins will have small pores. Okay, so the large proteins which will not be able to enter these small pores uh, will not enter into the small pores and will flow out faster. Okay, so these green proteins are not able to enter into these such small holes, so they will move rapidly through the column and be eluted fast, but the small proteins which are smaller in size will be able to enter the holes in this mesh like structure, and they will be retained for a longer period of time, which is why they will be released at a later, very later stage, and then they'll be eluted. Okay, so that is what happens in size exclusion chromatography or gel filtration chromatography or gel formation chromatography. Now, what is affinity chromatography? So, in affinity chromatography, the resin in the column will contain a covalently attached chemical group called a ligand. Okay, that is bound by the proteins of interest. Okay, so there is a ligand on the resin in the column, and the protein of interest that needs to be separated as a defined affinity towards that ligand. So when a mixture of proteins containing the protein that recognizes the ligand is applied to the column, other proteins will pass through, because obviously there is no attraction. So other proteins will pass through wash of the column, while the protein of interest will be retained, because that is a specific affinity towards the ligand. Okay, and the protein of interest will ultimately be eluted from the column by adding a buffer containing the free ligand. Okay, so if there is a free ligand, the free ligand will go and bind huge quantity of free ligand. The free ligand will bind to the protein of interest, and then they'll be eluted. So the free ligand will basically compete with the bound ligand for binding to the protein of interest, and the protein will dissociate from the resin. So very often affinity chromatography gives the largest fold purification of any step used in protein purification. So mostly it's the most primary and special way of purifying protein. So the first step will definitely be affinity chromatography for separating uh, pure protein from a mixture. Now, what is high performance liquid chromatography? It's basically the elution buffer is pumped over the resin at high pressure and speed. Okay, and this reduces the transit time of the protein on the column and improves the resolution of separation by reducing diffusional spreading of protein bands during the elution. So for purification tables, the progress of purification of a protein of interest is recorded in the purification table, and obviously the mass of total protein recovered is reduced while the specific activity of the protein fraction becomes greater. So this is a purification table for a hypothetical enzyme. For example, if you have an enzyme, we'll find out these many techniques, these many chromatography techniques to basically separate the fractional amount and the total protein, and check the activity as well. So anybody who didn't understand till this point, I'll repeat again. If you didn't understand, let me know. I'll give you 2 minutes. Anybody? 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 Okay, so we'll move on to SDSPA, the sodium dodecyl sulfate. Sodium dodecyl sulfate polyacrylamide gel electrophoresis is basically a routinely applied analytical method that is used to estimate the purity and the molecular weight of proteins in a sample. Okay, so basically the separation is based on the means of molecular weight and also purity, because we'll be able to see the bands. you'll be able to see if what we have gotten is a pure band or there are multiple bands. So proteins are dissolved in a buffer containing SGS and separated on a polyacrylamide gel matrix by the application of an electrical field. Okay, and the locations of the proteins on the gel after electrophoretic separation is performed is determined by staining the gel. Okay, determined by staining with the protein reactive dye such as Kumasi blue. And the figure below shows an application whereas SDS page was used to monitor the status of the purification of the rec A protein of E. coli. So this is how the sample preparation takes place, and this is how the setup looks like, and this is how you can monitor the purification of proteins from a complex cell lysate. Okay. So in SDS page, proteins are uniformly coated by SDS with about 1 molecule of SDS being bound per amino acid residue. And since SDS is strongly negatively charged under the electrophoresis buffer conditions, all proteins will adopt a roughly equal charge to mass ratio. So they further all adopt a rod-shaped structure in solution. The separation occurs based solely on molecular weight, with smaller proteins moving faster than larger proteins through the sieving matrix or the sieving the pores in the matrix of the gel. So the molecular weight of an unknown protein can be determined by running it on the gel in parallel with the known molecular weight standard, which we call ladder. Okay, any questions on SDS page? Okay, so later we come to isoelectric focusing. So what happens in isoelectric focusing is that proteins are separated according to the isoelectric points. Okay, so a pH gradient is established on a gel strip by allowing a mixture of low molecular weight organic acids and bases or ampholites to distribute themselves in an electric field applied across a strip, and proteins migrate on the strip to a point where the pH matches their exact PIs. So at the PI, the net charge on a protein is 0 and it stops moving in the electric field. So once it reaches its PI, it will stop because it has no negative charge right now, and it will stop moving in the electric field. Okay. So if you can see the diagram of a here, protein sample may be applied to one end of the gel strip with an immobilized pH gradient, or a protein sample in a solution of ampholites may be used to rehydrate a dehydrated gel strip. And electric field is applied, and you can see different bands. Anyone have any question on the last thing that we talked about? Isoelectric focusing. Okay, let's move on to two-dimensional electrophoresis. So, in two-dimensional electrophoresis, what happens is that isoelectric focusing and SDS page will be combined to obtain a high-resolution separation of a complex mixture of proteins. Okay, so what happens is that proteins are first separated by isoelectric focusing on a gel strip. Then the gel strip is mounted on top of an SDS page gel and separated by electrophoresis in the second dimension. Then horizontal separation of spores is based on their isoelectric point differences. So vertical supernatant is based on the differences in molecular weights. Thousands of proteins can be separated by this technique using a single SDS page gel. However, individual protein spots can be excised from the gel and identified by mass spectrometry. Now, what is activity versus specific activity? You come across these terms quite frequently when you start learning about proteins and enzymes. So, the purification of an enzyme is measured or assayed based on the ability of the protein fraction controlling the enzyme to carry out a biochemical reaction. For example, one unit of enzyme activity is defined as the amount of enzyme causing the transformation of 1 micromole of substrate to product per minute at 25 degrees Celsius under optimal conditions of measurement. So the term activity reflects the total number of specific. So the term activity reflects total units of enzyme in a solution, whereas the term specific activity is the number of units of enzyme activity per milligram of total protein. Very important, per milligram of total protein. The specific activity is a measure of enzyme purity. So it increases during the purification of an enzyme and becomes maximal and constant when the enzyme is pure. Okay. Now the levels of structures in proteins. So we have talked about the structure of large molecules. Such proteins can be taken or described at 4 levels of complexity, right? So they can be arranged in primary, secondary, tertiary, and quaternary stages in a conceptual hierarchy, basically. So the primary structure is nothing but the amino acid sequence of the protein, and it will include the disulfide bonds as well. Okay. The secondary structure will refer to the total local and stable folding elements in the larger structure, such as alpha helices, beta sheets, etc. Okay, and the tertiary structure will refer to the three-dimensional folding and locations of all atoms in the protein. So, quaternary structure is reserved for multi-subunit proteins, and the arrangement in 3D space of all subunits in a protein is its quaternary structure. So the first protein whose sequence was determined was that of bovine insulin. So the British Nobel laureate Frederick Sanger oversaw the sequencing of bovine insulin in 1953, the same year that Watson and Crick solved the structure of double helical DNA. Now today few protein sequences are determined in their entirety by chemical methods, with most being deduced from the DNA sequences of the genes that encode them. However, segments of proteins are often sequenced in the process of a gene cloning, and today in the study and many of the classical steps used in protein sequencing are applied today in the study of protein structure and function. So in this slide we'll talk about the modern methods for protein sequencing by chemical procedures, and mass spectrometry can also be discussed. So this is the basic chain length of these two amino acid chains. So what is the overview of protein sequencing? So the conventional strategy used for chemical sequencing of protein is summarized in this image. Okay, so it includes identification of the first residue located at the amino terminus, determination of the complete amino acid composition of the protein, cleavage of the protein into shorter polypeptide fragments that can be sequenced in their entirety, and fourth, sequencing of each polypeptide fragment, and fifth, ordering the polypeptide fragments in the overall sequence of the protein. So each of these steps is covered further in the next few coming slides. So if you see, we have to determine the amino terminus because it can react with FDNB, and glycine is there at the amino terminus. Then you have to determine amino acid consent by acid hydrolysis. Okay. Then cleaving to smaller polypeptides is trypsin, sequence of the trypsin, then determine order of the polypeptides in protein. Like peptide 3 will be at the order at amino terminus, peptide 2 will be at the carboxyl terminus. So you can see if this works out, and or rather say overlaps in sequence of peptides contained by leaving the protein with a different reagent, cyanogen bromide or trimotrypsin. Now, what is N-terminal labeling experiments reagents? The first amino acid residue in the N-terminus of a protein is defined by reaction of the protein with one of the reagents such as FDNB or 1,4,2,4-nitrobenzene. Okay, it is shown over here, FDNB. Now, following the label, the protein is hydrolyzed and the modified N-terminal residue is identified by chromatography. Okay. Now, this particular molecule is also known as Sanger's reagent. So, other sensitive related fluorescent reagents such as dancel chloride and dapsol chloride now are used when working with small quantities of a purified protein. Okay, so N-terminal labeling was also identifies which sequence polypeptide occurs first in the complete sequence of the protein. So that's what we'll get to know about these labeling reagents. Before we start, if there's any doubt, we can address it or we'll move on. I'll give you 2 minutes. Find out. Find out. Okay, so what happens in the Edmond degradation sequencing? So it's basically a chemical sequencing process based on a two-step procedure developed by Per Edmond. Okay, so the reagent phenyl isothiocyanate is used to label the N-terminal residue of the polypeptide, which then is released and identified without damaging the remainder of the polypeptide. Okay, then the process is repeated. About 40 amino acids can be sequenced for each polypeptide, and polypeptides themselves are generated by enzymatic or chemical fragmentation of the starting protein. Okay, so that is how it is seen. You can see this is the protein, it is the polypeptide. Then at high pH we'll add phenyl isothiocyanate. Okay, and then it will become phenyl thiocarbamyl derivative. Then again at low pH it will become anilino-thiazolinone derivative amino acid residue. Again at low pH it will form this residue. From here identifying amino acid terminal residue of the polypeptide can be done. And then you can, the peptide is shorter now. Now you can purify and recycle the remaining peptide fragments through Edmond process. Okay, that is how it happens. Now, how do we break disulfide bonds in proteins? So, in order to obtain isolated polypeptide fragments from protein for sequencing, disulfide bonds must be broken. So, commonly bonds are broken by oxidation with performic acid or using a two-step procedure in which the bond is first broken by treatment with the reducing agent such as dithiothretol, and then the free thiols are alkylated by carboxymethylation using iodoacetate. Okay, so this is a disulfide bond. Okay, so there will be oxidation with performic acid and there will be reduction by thiothretol. Okay, so if you see the reduction basically reduces your disulfide linkages. Okay, and then carboxymethylation is done by iodoacetate. So this is how a carboxymethylated cysteine residues will be seen. So what are the methods for fragmenting proteins? Since at most 40 amino acids at a time can be sequenced by the Edmond degradation process, large proteins must be fragmented into smaller polypeptides for sequencing. And a number of proteases are available for this purpose, as are a few chemical cleavage methods. Okay, like with the help of cyanogen, bromide cleavage, etc. So the selection of a method for cleavage is guided in part by the early determination of the total amino acid composition of the protein. So the identification of the C-terminal residue in the polypeptide fragments will help in ordering the polypeptides in the overall sequence. So the ordering of the protein sequence also is facilitated by sequencing in parallel a second set of polypeptides generated by another protease. So the order in which the fragments will appear in the original protein can then be determined by examining the overlaps in sequence between the two sets of fragments. Okay, that is basically the method for fragmenting the proteins. And this table shows the specificity of some common methods for fragmenting polypeptide chains. Now, what information can be derived from the protein sequence and analysis steps? So, the analysis of protein sequences via computer-based tools or bioinformatics will provide a huge, information about the structure and function of proteins. Obviously, if you're going to know about the sequence of proteins, then that will be able to tell you the specific amino acids that are there in the protein at which spots. That will be able to tell you which type of amino acids they are. Are they polar, non-polar, hydrophobic, what kind? And from there you can find out the folding of the proteins and how they interact with other partners and everything. Okay, so we can find out the evolutionary relationships between the organisms and can synthesize them as well. So a key method in the application of bioinformatics to evolutionary structure and function analysis is sequence alignments. The sequence alignment basically align you take two sequences and then you try to align them to see if there are similarities. The sequence alignments of related proteins also reveal consensus sequences that indicate protein function, cell location, chemical modification, and prosthetic group binding, and even turnover rates. Okay. Now about bacterial evolutionary trees, evolutionary relationships between organisms are revealed by comparing the sequences of fundamental proteins that are present in all organisms. So bacterial evolutionary tree derived from the comparison of the sequences of the protein GROW-L, which is a chaperone involved in assisted protein folding, is shown over here. In such trees, external nodes mark the locations of extant organisms, and internal nodes will mark the locations of their last common ancestors. Okay, so the lengths of the lines represent the level of sequence divergence between the organisms, and we can have to know that the comparisons of protein sequence are more reliable than the comparisons of DNA sequences from trees construction, since proteins contain 20 amino acids and DNA contains only 4 bases. So it's obviously more reliable to compare amino acid sequence, protein sequence, and not DNA sequence, because there are only 4 bases which give rise to so many amino acids and so many variations. Okay, but there are 20 amino acids, so it's better to compare the protein sequence. So sequence comparisons of numerous shared proteins and analysis of additional genomic features has been performed to construct the most likely general tree of life. Okay, such analysis revealed that all life forms on Earth can be divided into 3 domains: bacteria, archaea, and eukaryotes. So the evolutionary distances of each kingdom to the last universal common ancestor are given by the lengths of these lines in the tree. Okay. Any doubt till now, or we'll continue? So, why do we analyze proteins? Now, proteins play crucial roles in nearly all biological processes. Okay. Now, these many functions of proteins are a result of the folding of the proteins into many distinct 3D structures. So, different foldings will give rise to different structures, and different structures will be involved in different functions. As simple as that. Now, protein analysis tries to explore how amino acid sequences will specify the structure of proteins and how these proteins will bind to substrates and other molecules to perform their functions. Okay. So, protein analysis allows us basically to understand the function of the protein based on its structure. So, what are the main steps in protein analysis? Obviously, the extraction of proteins, the purification of protein, the structural characterization of the protein. That's how it's done. First, you overexpress the proteins, then you extract the proteins, then you purify, then you structurally characterize the protein. Now, proteins can be purified according to certain properties they possess. So, these properties allow us to apply different techniques in purifying proteins. We can think about the property of solubility. We can take the technique differential precipitation or molecular size, then gel filtration chromatography, size exclusion chromatography, gel permeation chromatography. Or if we are talking about molecular charge, we can go ahead with ion exchange chromatography. If we are talking about hydrophobicity property, then we can go with reverse phased HPLC. And if we are talking about the biological activity, we can talk about affinity chromatography. Why biological activity and affinity chromatography are matching over here? Can anyone say? Anyone? Okay, so we are talking about linking biological activity to affinity chromatography because in case of affinity chromatography, we have a ligand which will bind, we will have an affinity towards the protein of interest, right? So that is a biological activity, having another component that will have a specific affinity towards our protein of interest. Now, chromatography, as we talked, basically involves the separation of mixtures due to the differences in the distribution coefficient of sample components between different phases. And one of these phases can be called the mobile phase, and another will be called the stationary phase. Okay. So gel filtration, we have already talked about this in details. So it's a mechanical sorting of molecules based on the size of the molecules in the solution. Correct, and small molecules will be able to permeate more pores and are therefore retained longer than the large molecules. So how it works, a column will be packed with a semi-porous material, glass beads or glass wool, and something that doesn't dissolve in the solvent carrying the polymer chain. And in some cases, even simple soap detergents like Tide can be also used. Okay, so there will be a hydrodynamic radius. So if you see the column over here, protein mixture will be added to the column containing the cross-linked polymer, and the protein molecules will separate by size. Larger molecules will pass more freely, appearing in the earlier fractions. So the smaller molecules will get inside the pores, will travel longer distance, and will appear late in the later fractions. So chromatography is used to make reasonable estimates of the molecular weight distribution of polymers, and it should have be, if the work needs to be reproducible, you should have control over the flow rate, temperature, solvent composition, tunnel size of the stationary particles, and etc. Okay, this is the matrix types, different types of material that is used for extrusion chromatography. Solvents can be polar solvents like water with ethanol, isonitrile ethanol, oxypropionitrile, or non-polar solvents like cyclohexane and pentane, hexane, and decane. And detectors will be the UV detector and the refractive index RI detector. So increasing the column length, decreasing the column diameter, decreasing flow rate, packing column uniformly using uniform stationary phase, decreasing sample size, selecting proper stationary phase, proper mobile phase, using proper pressure. All of these, gradient illusion, all of these are very important factors in increasing the resolution. There are different matrix types for different size dextrins. Then we can run the column, and then we can figure out the calibrate of the column with known standards, and then we can plot the value of the unknown. So gel filtration chromatography is also known as ion exclusion chromatography, gel permeation chromatography, or molecular sieve chromatography. Okay, so it separates molecules according to their size and shape. And stationary phase will consists of beads containing pores that span a relatively narrower size range. The smaller molecules will spend more time inside the beads than larger molecules and therefore elute later. After a large volume of the more mobile phase, has passed through the column. The gels that are used as molecular sieves can be cross-linked polymers. They are either unchanged and inert, like they don't bind or react with the matrix analyzed, or they can be three types of gels. They can be dextran, which is a homopolysaccharide of glucose residues. Is prepared with various degrees of cross-linking to control pore size. It's bought as dry beads. The beads swell when water is added, and the trade name is called Cefadex. Okay, it's mainly used for the separation of small peptides and globular proteins with small to average molecular mass. Then there are polyacrylamide, which gels are prepared by cross-linking acrylamide with N-methylene disacrylamide. Their pore size is determined by the degree of cross-linking, and separation of properties of polyacrylamide gels are mainly the same as those of dextrans. With agarose, which is basically polymers of D-galactose and 3-cyanogalactose, it forms a gel that holds together with H bonds, hydrogen bonds, is dissolved in boiling water and forms a gel when it's cooled. The concentration of the material in the gel will determine the pore size. The pores of agarose are much larger than those of Cefadex or BioGLP. This is useful to and for analysis of separation of large globular proteins or long linear molecules such as DNA. Now, the gel filtration material that will be used in the experiment is basically mostly Cefadex. Okay, so it's basically for Cefadex column. The total volume is equal to the sum of the volume of the gel matrix and the volume inside the gel matrix and the volume outside the gel matrix. Okay. So, what are the advantages of gel filtration? It's the best method for separation of molecules differing in molecular weight. Doesn't depend on temperature, pH, ionic strength, and buffer composition. The separation can be carried out without any problem. There is very little adsorption. There is less zonal spreading than in other techniques, and the illusion of volume of retained after uh, the illusion volume is related to the molecular weight. So, what are the applications? Derivation of enzymes and other proteins, estimation of molecular weight mainly for globular proteins. So, this is how ion exchange chromatography works. If you can see the image over here, this is how gel filtration chromatography works. And all these techniques and ideas of isoelectric points, electric fields, very high resolution technique for protein, any kind of protein. So isoelectric focusing uses the theory of protein isoelectric point, which is basically a pH at which a given protein has a zero neutral overall charge. As neutral overall charge, zero positive or zero negative charge, or the equal positive and negative charge. The PI or the isoelectric point is dependent on which type of residues are present and how many. Okay, and it's specific for every protein. Okay, coming back to the question that we started with today, these will be the two statements that were correct. You have two statements which were correct. So, as we said, the gel-fission chromatography will separate molecules according to the size and shape, and the stationary phase will contain beads containing pores that span a relatively narrow size range. And smaller molecules will go inside the pores and will take more time inside the beads than the larger molecules. We have talked about these things. Okay. Okay, now next we'll come to the next question for today. Which of the following is true about the electrophoretic mobility mu of a protein? The ratio of The velocity of the protein to the electrical potential, the ratio of distance traveled by the protein and the electric potential, the ratio of molecular weight of the protein and the electric potential, functional coefficient, and the ratio of molecular weight of the protein and the electric potential. So you can look at the question for 2 minutes and think about an answer. We'll discuss after that. No, sorry, there's a no, sorry, there's a Sorry, there's a yes. So we'll continue talking about this question. So, which of the following is true about the electrophoretic mobility of a protein? Correct. So the ratio of the velocity of the protein to the electric potential, the ratio of the distance traveled by the protein and the electric potential, the ratio of the molecular weight of the protein and its frictional coefficient, and the ratio of the molecular weight of the protein and the electric potential. Okay, so the answer, we'll come back to the answer, but we'll discuss basically about the principle. So the easiest way to carry out protein electrophoresis in a gel is basically the separation results will be visualized by staining of these J's with dyes and specifically bind to these proteins. Okay. So electrophoresis of proteins in gels has become a widespread separation technique because it has a very high resolving power. It's fast. It's easy to perform in relatively simple instruments, and the consumables do not really cost that much. Okay, so each protein has a specific electrophoretic mobility, which will determine its migration velocity in an electric medium, and is therefore decisive for the separation. Okay, and the electrophoretic mobility is dependent on the net charge and the size of the molecules as well. So, proteins with different electrophoretic mobilities can migrate in the electric field with different migration velocities and different for distinct zones. So now proteins are amphoteric molecules. So they will have different net charges that depend on their pH environment. Okay. So this is a schematic of a net charge curve of a protein. The shape of the curve and its intersection with the x-axis describes the behavior of a protein at a certain buffer pH value. And in the electric field, it should be mentioned that the net charge curves of proteins can be determined by a simple to perform electrophoretic technique called titration curve analysis. It is very valuable for ion exchange chromatography. Okay, if you can see the curve, this is a schematic of a net charge curve of a protein. So protein with this net charge curve has an isoelectric point at pH 8 and two positive charges at pH 6 and one negative charge at pH 9. Now, there are several electrophoretic techniques, separation principles that can be applied on proteins. Moving boundary electrophoresis, isoelectric focusing, and zone electrophoresis. Okay, so if we talk about gels, polyacrylamide gels are predominantly used for protein electrophoresis. In some exceptional cases, agarose gels can also be used. So agarose gels are used for electrophoresis in clinical diagnostics and when large pools of the analysis of large proteins over 800 kDa are needed. So agarose is a polysaccharide obtained from red seaweed. Now polyacrylamide forms a much more restrictive gel and is mechanically and chemically more stable and has lower electroendosmosis and is clearer than agarose. This is a vertical SDA's polyacrylamide gel. The two lateral lanes are molecular weight markers. If you can see the molecular weight markers, and the other lanes are basically buffer extracts of legume seeds. So vertical slab gels are mostly polymerized between glass plates and lateral spacers, which determine gel thickness. Gels are left in these cassettes from casting to the end of the run, and the samples are then loaded in the slots on the top edge, okay, which have been formed by inserting a comb during polymerization. Now, combs with different thicknesses and numbers of teeth are available, obviously. Now, prefabricated gels are apparently also shipped in the cassette, including the inserted combs, and they are then inserted into the appropriate apparatus. The upper and lower gel edges are in direct contact with the electrode buffers. So, to prevent mixing of the samples with upper buffer, 20% glycerol or sucrose is added to the sample offered in order to increase the density of the samples markedly. The simplest design for vertical electrophoresis, the gels cannot be cooled. The applied electrophoretic field is therefore to be limited accordingly. Okay. So what can be the types of electrophoresis? There can be paper and cellulose electrophoresis, there can be thin electrophoresis, there can be pH, there can be agarose gel, and there can be capillary electrophoresis. So basically in this method, the polyacrylamide will be used as a support matrix. Acrylamide monomers covalently bond with cross-linker and N-prime methylene disacrylamide to form a polymer. The polymerization process will be done chemically or photochemically. In chemical methods, ammonium persulfate, and in photochemical methods, riboflavin is used. And these can act as free radical initiators. The formed radicals will allow the polyacrylamide molecules to be linked. Okay, so acrylamide and N prime methylene disacrylamide will come together, and then they will cross-link. So polyacrylamide polymers obtained from acrylamide monomer are obtained. So the porosity of the polyacrylamide can be changed by changing the acrylamide content. So pH method can be applied to the separate native proteins. This technique is also used to determine the molecular mass by denaturing the proteins using SDS. So SDS pH is a protein electrophoresis technique widely used for research in mol bio. And this technique is also a much better separation than routine use in clinical labs. Okay. It also provides separation of a large number of subunits of proteins. Now the gels used in gel electrophoresis according to the type of matrix can be starch, agarose, or polyacrylamide. So the matrix used in electrophoresis must be unloaded. If the matrix is loaded, the progression of the loaded biomolecules is prevented. So as the concentration of the substance in the matrix increases, the pore diameter becomes smaller. So according to the pore diameters, the largest porous gel is agarose, then starch, and then the smallest porous gel is polyacrylamide. Okay, 1 second. So there are obviously two different systems in electrophoresis, the continuous and discontinuous. So in the batch system, there is a second gel on the bottom gel. After the lower gel is polymerized, the upper gel is placed and the comb is placed. So after the comb is removed after completion of the polymerization, the voids are formed in the upper gel, and the upper gel loading gel is large pores and the lower gel separating gel has small pores. So the applied sample first passes through the big porous loading gel, then passes through the sub gel, and the separation is achieved. The two parts in the batch system are prepared at different concentrations and pH, and this technique is most widely used form of pH. So the continuous gel contains a single gel, but generally people use the discontinuous gel system. Okay, so this is schematic of vertical plate. This is how the pattern will look like, and from the ladder you can calibrate and make the standards, basically make the standard curve, and from that you can figure out the molecular weight of the unknown. So if we think about the transportation in electric field, then we have to talk about electrophoresis, because it requires a matrix, either agarose or polyacrylamide, to minimize convective effects due to heating and Brownian motion, and mobility is also affected by the partitioning within the stationary phase. It provides molecular weights determined by comparison to standards. Transportation in gravitational field requires centrifuge to generate the large gravitational forces required for protein transport, provides an absolute molecular weight and informs on the oligomerization state for stable complexes. If we talk about transport by partitioning between mobile and stationary phases, it requires a suitably sized partitioning matrix for the solid phase. It provides relative molecular weights determined by comparison to standards. So what is electrophoretic mobility is basically proportional to its net charge and especially the ratio of its net surface charge to the accessible surface area, and inversely proportional to its frictional coefficient, that is a function of solvent viscosity and protein geometry. So the constant of proportionality is unique to each protein. Okay, so mobility can be measured to the cathode or anode depending on the protein charge and pH. And free radicals are provided by ammonium persulfate. Temid or tetramethylene diamine is included to stabilize the free radicals resulting from decomposition of the ammonium persulfate, and the ratio of acrylamide to bisacrylamide is held constant. So pore size is expressed as percentage weight by volume acrylamide and designated as percentage T. So SDH is full form will be sodium tritisyl sulfate. So STS basically binds to the protein with a constant ratio of 1.4 gram STS per gram of protein, basically to impart a uniform negative charge to the proteins. We've also talked about gel filtration and or size exclusion chromatography. Okay, and how comparison of size resolution by gel filtration and pH will happen, and how the calibration plots for molecular weight standards result on different media can take place. Okay, moving on to the next question for today. Identify the options in which the dipeptides shown in column 2 are correctly matched with the amino acids in column 3. So you have three options. You have to do match and pair. Anyone? Anyone? Okay, so the answer will be A will match with alanine and glycine, B will match with proline and tryptophan and alanine, and C will match with tryptophan and lysine. So we have talked about amino acids, the building blocks of proteins. We have talked about the different amino acids and how at physiological pH range both carboxylic and amino groups are completely ionized. They can act as either an acid or a base. They are deuterions or molecules that have both a positive and negative charge. So because of the ionic nature, they have extremely high melting temperatures. Amino acids can form peptide bonds. So the amino acid residue, peptide units, dipeptides, tripeptides, and proteins that are molecules that consist of one or more polypeptide chains. And peptides are linear polymers that can range from 8 to 4,000 amino acid residues. So we've already talked about all these. What are the characteristics of amino acids? There are three main physical categories to categorize describe amino acids. Non-polar, hydrophobic, 9 in all, like glycine, alanine, valine, isoleucine, methionine, proline, and alanine, tryptophan. Uncharged, polar, 6 in all, serine, threonine, asparagine, glutamine, tyrosine, cysteine. Charged, polar, 5 in all, like glycine, arginine, glutamic acid, aspartic acid, and histidine. So amino acids, you must know their names, their structure, their one letter code, and the three letter codes. Okay, this is the table that will help you remember the same. Cysteine consists of disulfide linked cysteine residues. This is the structure of cysteine. If there is a dipeptide with two cysteine, this is how it will be looking like. Okay. We have also talked about in one of the previous classes about the acidic properties of amino acids. Isoelectric point is basically the pH at which a protein carries no net electrical charge. Okay. So we have talked about how break lettering is used to identify atoms in lysine or glutamate. Okay. So what are proteins? Basically, proteins are large molecules made up of chains of amino acids. Amino acid building block of proteins. They are found in every cell in the body. They're involved in most of the body's functions and life processes. The sequence of amino acids is determined by DNA. Structure of proteins is made up of chains of amino acids classified by number of amino acids in a chain. Okay, so peptides can be fewer than 50 amino acids, dipeptides 2 amino acids, tripeptides 3 amino acids, and proteins can be more than 50 amino acids. So chains are synthesized based on specific bodily DNA. The amino acids are composed of carbon, hydrogen, oxygen, nitrogen. Okay. Now, what are the structural differences between carbohydrates, lipids, and proteins? Carbohydrates are chains of glucose. Example, there will be glucose units. Lipids are chains of fatty acids, will be triglycerides, and proteins will be chains of amino acids. Okay. So this is the anatomy of an amino acid. This is the amino structure. We have the amine group, the acid group, and a side chain. Okay. So different amino acids will differ in their properties because they have different and unique side chains. Now the amino acids link the peptide bonds link amino acids and form when the acid group of one amino acid joins with the amine group of a second amino acid. So it's formed through condensation and broken through hydrolysis. So see, a peptide bond is formed by the condensation when the acid group of one amino acid and amine group of the other amino acid join and release a molecule of water. And when peptide bonds are broken by hydrolysis, the hydroxyl group and the hydrogen from water are added. So now we have essential, non-essential and conditional amino acids. Essential must be consumed in the diet. Non-essential can be synthesized in the body, so they are non-essential in the diet. And conditional essential, they cannot be synthesized due to illness or lack of necessary precursors. Okay. So this is the essential, these are the essential amino acids and the non-essential amino acids in a table format. The structure of the protein, we know about the four levels of structure: primary or secondary, tertiary, and quaternary. So any alteration in the structure or sequencing changes the shape and the function of the protein. Now what is denaturing? Alteration of the protein's shape and thus function through the use of heat, acids, base, salts, mechanical agitation, all of these. Primary structure is unchanged by denaturing. So even after denaturing, will change the folding of the structure you will basically denature the folding. You will not be able to do anything to the primary structure. That is the sequence of amino acids that cannot be changed even with denaturing. So this is how normal protein will look like. You can denature using these reagents and make the protein look like this. Now, proteins are chains of combination of amino acids. Amino acids contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. And unique amino acids will consist of a central carbon with a carboxyl group, a hydrogen, a nitrogen containing amine group, and a unique side chain. So there are 20 side chains and 20 unique amino acids. Okay, and amino acids link together with the peptide bonds by condensation and break apart by hydrolysis. Now, attractions and interactions between the side chains cause the proteins to fold into precise three-dimensional shapes. Protein shape determines function. Proteins are denatured and the shapes changed by heat, acid, bases, salts, and mechanical agitation. Okay, so that's what it happens. So if we go back to the question that we started with, so identify the options in which the dipeptide shown in column 2 are correctly matched with the amino acids in column 3. It will go with this one. A2, A will be alanine and glycine, B will be proline and phenylalanine, and C will be tryptophan and lysine. Okay, so that's the difference between the all different amino acids. They're structurally different because the side chain, the R group is different. Otherwise, all of these will contain amino group, a carboxyl group, but the R group is different. So, anybody has any questions till now? Okay. 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 Moving to the next question for today, which of the following compositions of enzymes is true? Apoenzyme plus holoenzyme is coenzyme. Holoenzyme plus coenzyme is apoenzyme. Coenzyme plus apoenzyme is holoenzyme, and coenzyme plus prosthetic group is holoenzyme. Anyone? The correct option is coenzyme plus apoenzyme is holoenzyme. Right? So the enzyme without its non-protein moiety is termed as apoenzyme. Correct? And it is inactive. And the holoenzyme is an active enzyme with its non-protein component. So apoenzyme plus the cofactor or the coenzyme will basically give rise to the whole enzyme, which is the holoenzyme. right? So the name of an enzyme in many cases will end in ASE, like sucrase will catalyze the hydrolysis of sucrose. Uh, Oxidase will catalyze the oxidation of reactions. So sometimes common names are used, particularly for the digestion enzymes such as pepsin and trypsin. And some names can describe both the substrate and the function. For example, alcohol dehydrogenase oxidizes ethanol. So, what is the mechanism of action of enzymes? So, enzymes will increase the reduction rates by decreasing the activation energy. So, enzyme substrate interactions will be basically the formation of enzyme substrate complex by lock and key model or induced fit model. Okay, so without enzyme, the activation energy will be huge, and net energy released from splitting of lactose will be this. But with the enzyme, the activation energy will be decreased. Okay. So in the lock and key model of enzyme action, the active site is a rigid shape. Only substrates with the matching shape can fit. The substrate is the key that fits the lock of the active site. This is an older model, however, and does not work for all enzymes. Okay. So lock and key model is the active site, and then we have the enzyme substrate complex. So the lock and key hypothesis basically shows the substrate interacting with the enzyme, formation of enzyme substrate complex, and then enzyme maybe will form the product, will release the product, and it can be used again. So enzyme specificity. The enzymes have varying degrees of specificity for substrates. So enzymes will recognize and catalyze a single substrate, absolute, a group of similar substrates like relative, and a particular type of bond. It's also relative. So there are three factors that can affect enzyme activity: environmental conditions, cofactors and coenzymes, or enzyme inhibitors. So environmental conditions like extreme temperatures can be very dangerous. High temperatures may denature the enzyme, and pH also. pH the enzymes will work good at their optimum pH or optimum temperature. And also substrate concentration is also very important. Whereas cofactors, coenzymes, inorganic substances as zinc and iron and vitamins respectively are sometimes needed for proper enzymatic activity. So, example, iron must be present in the quaternary structure hemoglobin in order for it to pick up oxygen. Optimum temperature, the temperature at which enzymatic reaction can occur the fastest, that will be the optimum temperature. Now, pH also affects the rate of enzyme substrate complexes. So, most enzymes have an optimum pH of around 7. However, some may prefer acidic or basic conditions. Like pepsin will prefer an acidic condition, whereas trypsin will prefer a little bit alkaline condition. Substrate concentration and reaction rate. So, rate of reaction increases as substrate concentration will increase at constant enzyme concentration. Maximum activity will occur when the enzyme is saturated, that is, all the enzymes are binding substrates. Okay. So, what are the general properties of enzymes? So, increased reaction rates will sometimes, like enzymes will not change the delta G, just the reaction rates. Okay. So, milder reaction conditions with associated and can be regulated. That are the general properties of enzymes. So, non-covalent bonds and forces are maximized to bind substrates with considerable specificity. There are van der Waals forces, electrostatic bonds, anionic interactions, hydrogen bonding, hydrophobic interaction, etc. Okay, so this is an enzyme, this is substrate. Enzymes are stereospecific. Okay, so if now what are coenzymes? Coenzymes basically are smaller molecules that aid in enzyme chemistry. So enzymes can carry out acid-base reactions, can help in transient covalent bonds. Okay, charge-charge interactions. Enzymes cannot do oxidation reduction reactions or carbon group transfers. But the prosthetic group, permanently associated with an enzyme or transiently associated. And our enzyme will be catalytically active enzyme with cofactor, and apoenzyme will be enzyme without its cofactor. So these are common coenzymes, and what are the reactions that they mediate. Vitamins are useful coenzyme precursors. So there are two general ways to control enzymatic activity: to control the amount or availability of the enzyme, and to control or regulate the enzyme's catalytic activity. Okay. So enzyme amounts in a cell can depend upon the rate in which it is synthesized and the rate it is degraded. Now, synthesis rates can be transcriptionally or translationally controlled, and degradation rates of proteins can also be controlled. So the catalytic activity of an enzyme can be altered either positively or negatively through conformational alterations or structural modifications. Okay. So also substrate binding to the enzyme may also be modified by small molecule effectors changing its catalytic site. So protein phosphorylation of serine residues can activate or deactivate enzymes. So these are generally hormonally controlled to ensure a concerted effect on all tissues and cells. So that's why if we go back to the question we started with, it will be apoenzyme plus coenzyme will give rise to holoenzyme. That was the question, and that is exactly what we reach at the end of this discussion. Apoenzyme, coenzyme plus apoenzyme will be holoenzyme. So that is the correct answer. So this was the last question for today. Any of you have any doubt regarding these questions, or we can conclude the session for today and meet again next Monday. Anybody? Any question?